Okay, we are going to look at the rate of photosynthesis today. This is following on from last week's lesson on the uses of glucose. By the end of this session, you are expected to do the following three things. Firstly, identify factors to change, measure, and control to test a hypothesis. Second, collect and display data appropriately. And finally, draw conclusions from data collected. Before we start, there are a few things that we need to have in place so you can get the most out of this session. Number one, turn off your phone or put it in a different room. Number two, don't have any other tabs open on your computer. Number three, answer all the questions. If you need to, pause the video when you can. Number four, you can always rewind and listen again if you are not sure. And number five, don't give up. Now, to kick things off, you need to complete the exam question. So pause the video and do the exam question now. Using a green pen, mark your work. Pause the video and self-assess. So here is the equation for photosynthesis. Carbon dioxide reacts with water in the chloroplast of cells. Using light energy, it converts into glucose and oxygen. Now the key idea here is, or question is, how can we measure how quickly photosynthesis is happening? What are your thoughts on this? One of the things you could do is actually measure the amount of glucose being produced per unit time. However, that would be rather difficult. Another thing you could do is see or measure how quickly a plant grows within a lesson. Again, that would be very difficult because a plant, it will grow, but the growth would not be noticeable. The final thing you can do is you can measure the amount of oxygen produced by the plant if you have a plant which naturally inhabits a water environment. Now there are three factors that affect how quickly photosynthesis takes place. Firstly, there's light intensity. So the more light you have, the more energy there is to transfer and use for reacting glucose and water. The temperature. The higher the temperature, the quicker the reaction because the glucose and water will collide with each other more often. And the amount of carbon dioxide levels. Remember, for photosynthesis to take place, one of the reactants needed is carbon dioxide. So the more carbon dioxide you have, the more photosynthesis you'll end up with. On this slide is the basic setup for measuring the amount of gas or oxygen produced by a photosynthesizing plant. So what you will need is a beaker which is filled with water. You'll need an inverted test tube which is also filled up with water. You need a funnel with your plant inside. Now what will happen is that over time, as the plant is photosynthesizing, oxygen gas will be produced, and this oxygen gas, obviously it's lighter, less dense than water around it, will go up into the test tube, displacing water there. What you can do here is, afterwards, to test for water, you can do what we call the relighting glowing splint test. So get a splint, light it up, blow it out, so it's glowing, and we quickly put it into the gas that you've collected. If that splint relights, then that proves you have oxygen gas. On this slide, what you need to do is look at the diagram very carefully, okay, and decide which of these things, or what is the independent variable, and give a range and interval for this. So don't forget, the independent variable is the thing that you change in an experiment. You then need to identify the dependent variable. So what are you measuring? And finally, you need to identify the control variables. So for each experiment, what do you need to keep the same so the experiment is as fair as possible? Now using your green pen, assess your work. Now on this slide you have a link to a YouTube video. Now watch this YouTube video. It's a video or a clip of the actual practice itself, hopefully reinforcing everything we have looked at on the last two slides. Now on the graph paper in your booklet, you need to do the following thing. You need to plot the table results into a graphical format. So a few things you'll need to consider is, where do you place your axis? How big is your scale going to be? And what kind of line of best fit does the data show? Is it a curved line or a straight line? Okay, if you look at the slide, this is what your graph should have looked like. You should have both axes complete with their labels and units. The scale should be evenly spaced. The plots should be correct, and your line of best fit is actually a curve. Now using your graph and the data provided, you need to write a conclusion. In your conclusion, describe the pattern that is shown by the results. Describe what is the data that backs up the description that you've given. Explain why we see this pattern. And finally, you should have made a hypothesis earlier, so do your results support the hypothesis? You have got 10 minutes to complete this task. Using your green pen, assess your work. So light intensity is a factor which can increase or decrease the rate of photosynthesis depending on the amount you have. So increasing light intensity will increase the rate of photosynthesis, but only up to a point. Okay? Beyond this point, even if you increase the intensity, the rate of photosynthesis will remain constant. And that is because there are other factors which also affect the rate of photosynthesis. So these include your carbon dioxide and temperature. A limiting factor is the factor that is in the shorter supply for the plant. Okay, so this is the factor which slows down or limits the rate of photosynthesis. Now, any of these variables can be a limiting factor. It could be light intensity, the availability of carbon dioxide, or temperature itself. You need to check your understanding, so I need you to answer these three questions. The first question is, which factor is most likely to be limiting photosynthesis on a frosty, sunny morning? Number 2, which factor is most likely to be limiting photosynthesis on a cloudy summer day? And finally, question 3, which factor is most likely to be limiting photosynthesis inside a greenhouse? Okay, using green pen, assess your answers. To finish this session off, you need to complete the exam questions in the booklet. Okay, well done.